Let us begin by settling the mind. The body you inhabit is quietly alive with movement and coordination, even when you are still beneath the surface. Complex systems work together with remarkable consistency, sustaining your life without demanding your attention. One of these is the digestive system, a continuous passageway, assisted by specialized organs that receives what you eat and transforms it into the energy and building materials your body needs. In this gentle opening, we will take a broad look at the digestive system as a whole. We will see it not as a set of isolated parts, but as one long continuous process that begins when food first enters the mouth and concludes when the remains are released from the body. It is a system of movement, transformation, and absorption unfolding in an unbroken rhythm. The digestive system stretches from the oral cavity, through the throat, down the esophagus, into the stomach, through the small and large intestines, and finally to the anal canal. Alongside this main tract are accessory organs, the liver, gallbladder, and pancreas, which connect by ducts and channels, providing essential secretions that assist in digestion. Though they do not directly carry food, their contributions are indispensable to the process. We can think of the system's primary tasks as twofold. The first is the breakdown of food into smaller, absorbable units. This occurs through both mechanical digestion, the physical processes that crush and mix, and chemical digestion, the use of enzymes and other substances to break down complex molecules into simpler ones. The second major task is the absorption of those simpler molecules into the bloodstream or lymph, allowing them to travel to cells throughout the body. Everything else that cannot be used is eventually prepared for elimination. As we trace the digestive pathway in our minds, we will notice that movement is not rushed. It is paced by the steady wave-like contractions of smooth muscle known as peristalsis. These movements occur throughout the digestive tract, gently pushing food forward while also mixing it with secretions. This rhythmic motion is constant, even while you sleep, guided by built-in patterns in the muscular walls of the tract. The digestive process begins in the mouth, where food is taken in, chewed, and moistened with saliva. The chewed portion, now called a bolus, is swallowed and guided through the pharynx and esophagus toward the stomach. The stomach serves as a reservoir and mixer, churning the food with gastric secretions to produce a semi-fluid mixture called chyme. From there, the chyme is released in small amounts into the small intestine, where the majority of chemical digestion and absorption takes place. Here, bile from the liver and gallbladder and enzymes from the pancreas meet the chyme, breaking it down into molecules the body can absorb. The lining of the small intestine is covered with folds, villi, and microvilli, structures that increase surface area, allowing for efficient absorption of nutrients into the blood or lymph. What remains then passes into the large intestine, where water and electrolytes are absorbed, and the material is compacted into feces. The large intestine also houses a diverse population of microorganisms, known collectively as the gut microbiota, which play an important role in breaking down certain substances, producing vitamins, and supporting immune function. This entire sequence from ingestion to elimination is coordinated by a combination of nervous system control and hormonal signals. The enteric nervous system, sometimes called the second brain, resides within the walls of the digestive tract, regulating movement and secretion without requiring conscious thought. Hormones such as gastrin, secretin, and cholecystokinin adjust the timing and activity of different digestive organs to ensure the process flows smoothly. It is important to remember that digestion is not a series of isolated steps, but a continuous journey. The stomach does not work alone, nor does the small intestine function in isolation. Instead, each part is dependent on what came before, and each step prepares the way for the next. The secretions of the liver are useless without the mixing actions of the stomach. The absorptive surface of the small intestine would be overwhelmed without the regulated release of chyme through the pyloric sphincter. Even the act of swallowing is connected to digestion much further along. The pace at which food moves through the tract can influence how much water is absorbed in the colon or how quickly hunger returns. These subtle interconnections mean that the digestive system is best understood as a single integrated whole, a living corridor that transforms what we consume into what we are made of. For the purposes of study, we often divide the digestive tract into segments. The alimentary canal refers to the main passageway itself: mouth, pharynx, esophagus, stomach, small intestine, large intestine, rectum, and anal canal. The accessory organs include the teeth and tongue in the mouth, as well as the salivary glands, liver, gallbladder, and pancreas. But these divisions are organizational tools. In function, the system operates as one continuous process. Over the next sections, we will explore each part in greater detail, but for now, the overview is enough. We will carry forward the idea that digestion is not hurried and not fragmented. It is a calm and deliberate sequence, carried out by specialized structures working in harmony. As you listen, you may notice certain terms returning. This gentle repetition is intentional. In the same way that the body repeats the rhythm of peristalsis, learning is often helped by cycles of review. Words like chyme, peristalsis, and absorption will become more familiar each time they appear, without pressure to memorize them at once. Even complex ideas such as the difference between mechanical and chemical digestion can be absorbed gradually. Mechanical digestion includes the physical breakdown of food, chewing, mixing, and churning. Chemical digestion involves breaking down large molecules, proteins, carbohydrates, fats, into their smaller building blocks using enzymes and other chemicals. Both are essential, and both are happening at different stages along the tract. When you consider the digestive system in its entirety, it is easy to see why it is so fundamental to life. Without it, the energy stored in food could not be accessed. Without absorption, the building blocks needed for growth and repair could not reach the cells. And without the orderly elimination of waste, the body's internal environment could not remain stable. This overview is our first step. A slow, calm introduction to the landscape we will soon explore more closely. In the next section, we will begin at the very start of the journey, in the mouth, where digestion quietly begins before we even think of it as such. For now, let this picture of the whole system settle in your mind, a single, unbroken pathway, aided by silent helpers, working at a pace that is steady and sure. We now arrive at the very beginning of the digestive pathway, the oral cavity, or mouth. It is here that the first contact between food and the digestive system occurs, and though this stage may seem simple, it is a place of remarkable coordination between structure, sensation, and function. The mouth is framed at the front by the lips, which help guide food inward, and bordered on either side by the cheeks, which assist in keeping food between the teeth during chewing. Inside, the cavity is divided into two regions. The vestibule is the space between the lips or cheeks and the teeth, while the oral cavity proper lies within the arch of the teeth, extending back toward the throat. The teeth themselves are specialized tools for mechanical digestion, the physical breaking down of food into smaller fragments. Humans have two sets in a lifetime: the deciduous or baby teeth, which are eventually replaced by permanent teeth. Each type has a role. The incisors at the front cut food into pieces. The pointed canines tear and pierce. The premolars and molars crush and grind, turning larger pieces into manageable fragments. This process of chewing, known as mastication, increases the surface area of food, making it easier for digestive enzymes to work effectively in later stages. Above the teeth lies the hard palate, forming the bony roof of the mouth, which separates the oral cavity from the nasal passages above, toward the back. This firm structure gives way to the soft palate, a muscular fold that ends in the small conical projection called the uvula. The soft palate plays a role in swallowing, helping to close off the nasal passage so that food is directed downward toward the pharynx. The tongue rests on the floor of the mouth, anchored by a fold of tissue called the frenulum. This muscular organ is far more than a passive surface. It moves food around during chewing, presses it against the teeth, mixes it with saliva, and shapes it into a rounded mass, the bolus, ready for swallowing. The upper surface of the tongue is covered with tiny projections called papillae, some of which house taste buds. These sensory structures detect the basic tastes: sweet, sour, salty, bitter, and umami. In this way, the mouth is not only the mechanical entry point for digestion, but also an early center for sensory evaluation, guiding us toward what is safe and nourishing. Alongside mechanical breakdown, chemical digestion begins here as well. This is made possible by saliva, a clear fluid produced by the salivary glands. Three major pairs of salivary glands contribute: the parotid glands located near the ears, the submandibular glands beneath the jaw, and the sublingual glands under the tongue. Smaller minor salivary glands in the lining of the mouth add further secretions. Saliva is mostly water, which moistens food, making it easier to chew and swallow, but it also contains important components for digestion and protection. Salivary amylase begins the chemical breakdown of starches into simpler sugars. Lingual lipase, secreted in small amounts, can begin the digestion of fats, though it is more active in the stomach. Saliva also contains mucus, which lubricates the bolus, and antimicrobial substances such as lysozyme and antibodies that help protect the mouth from harmful microorganisms. The act of chewing and the presence of food in the mouth stimulates salivary secretion. This process is controlled by the autonomic nervous system, with parasympathetic signals promoting a steady, watery flow of saliva. Even the thought, sight, or smell of food can trigger this reflex, preparing the mouth for incoming nourishment. The sensory experience of eating begins here as well. The texture of food is detected by touch receptors in the oral tissues. Temperature is sensed by thermoreceptors. Smell, though technically a function of the olfactory system, combines with taste to create the perception of flavor. These inputs travel to the brain, where they are integrated into a single impression of the food, influencing both enjoyment and digestion. The mouth is also where digestion connects closely with the respiratory system. During breathing, air passes through the mouth and into the pharynx, sharing space with the pathway for food. The careful separation of these functions during swallowing is essential, and the structures of the mouth and throat are designed to ensure that food is directed safely toward the esophagus rather than the airway. When chewing is complete, the tongue gently pushes the bolus toward the back of the mouth, initiating the swallowing reflex. At this point, control shifts from voluntary to involuntary as muscles in the pharynx take over. The mouth has completed its role in preparing the bolus, and the process continues deeper into the digestive tract. It is worth pausing to recognize that the mouth is not simply an entry gate, but a finely tuned environment. Every structure, from the enamel-covered crown of a tooth to the microscopic taste receptor within a papilla, is specialized for its role. Mechanical and chemical digestion begin here in seamless partnership, setting the stage for all that follows. For the purposes of study, the functions of the mouth in digestion can be summarized softly. It receives and positions food. It breaks food into smaller pieces mechanically. It mixes food with saliva, initiating chemical digestion of starches. It forms the bolus for safe swallowing. It engages multiple senses to assess and prepare for further digestion. These processes occur without urgency, often without conscious thought, yet they are essential for the body's use of nutrients. By the time food leaves the mouth, it has been physically reduced, chemically altered, moistened, and evaluated, a quiet but comprehensive preparation. In later stages, enzymes in the stomach and small intestine will take over most of the chemical work, and the muscular contractions of the gut will replace the chewing motion. But these later processes depend on the efficiency and completeness of what happened here, at the very beginning. Poor chewing or inadequate mixing with saliva can make digestion less effective further along. As we move forward in our exploration, keep in mind that every stage of digestion builds on the one before, the mouth. With its combination of mechanical skill, chemical initiation, and sensory integration, creates the ideal starting point for the long journey of food through the body. Now that we have lingered here at the gateway, Observing its structures and appreciating its quiet precision, we are ready to follow the bolus into the next stage. In the section ahead, we will see how swallowing carries food from the mouth into the pharynx, and how the body's reflexes protect the airway while guiding the bolus safely into the esophagus. For clarity, the pharynx is divided into three regions. The nasopharynx lies behind the nasal cavity and is primarily involved in breathing. Below it is the oropharynx, situated directly behind the oral cavity. This is where the bolus enters after leaving the mouth. Further down is the laryngopharynx, which connects both to the esophagus, leading to the stomach, and to the larynx, leading to the trachea and lungs. With the bolus now prepared in the mouth, softened, moistened, and shaped by the tongue, it is ready to begin its journey deeper into the body. The next stage is swallowing, a process so familiar and effortless that we rarely notice it. Yet beneath the surface, swallowing is a carefully timed sequence involving a shared space that serves both digestion and breathing. The pharynx is that shared region. It lies behind the mouth and nasal cavities, connecting upward to the nasal passages and downward to both the esophagus and the trachea. In digestion, it serves as a short but crucial passageway for food and liquid on their way to the esophagus. In respiration, it carries air toward the lungs. This dual role means that swallowing must be precisely coordinated to ensure that food is directed toward the esophagus and not into the airway. The pharyngeal phase begins as sensory receptors in the oropharynx detect the presence of the bolus. Signals travel to the swallowing center in the medulla oblongata and pons of the brainstem. In response, a sequence of muscular actions unfolds almost instantly, though we will describe it slowly. The soft palate rises, closing off the nasopharynx so that food cannot enter the nasal cavity. At the same time, the larynx moves upward, and the epiglottis, a flexible flap of cartilage, tilts downward to cover the laryngeal opening. This simple but essential motion directs the bolus away from the airway and toward the esophagus. Swallowing or deglutition can be described in three phases: the voluntary phase, the pharyngeal phase, and the esophageal phase. Only the first is under conscious control. The rest are guided by reflexes, allowing the process to occur smoothly and safely without the need for active thought. In the voluntary phase, the tongue presses the bolus against the hard palate and then pushes it toward the back of the mouth. This movement propels the bolus into the oropharynx, marking the point at which the swallowing reflex is triggered. From this moment forward, the process becomes involuntary. During this phase, breathing is momentarily paused. This brief suspension is so natural that it goes unnoticed, yet it is critical to prevent inhalation of food or liquid. The coordinated contraction of the pharyngeal muscles gently squeezes the bolus downward into the laryngopharynx, ready to enter the esophagus. The esophageal phase then begins as the upper esophageal sphincter, a ring of muscle at the top of the esophagus, relaxes, allowing the bolus to pass. Once inside, peristaltic waves take over, moving the bolus toward the stomach. This final phase will be explored in more detail when we follow the esophagus itself in the next section. Though the swallowing reflex happens quickly, it is supported by a remarkable choreography of muscles, nerves, and protective structures. More than 20 pairs of muscles are involved, working in coordinated patterns controlled by both cranial nerves and the brainstem. The trigeminal nerve, cranial nerve V, helps control chewing muscles, while the facial nerve, cranial nerve V, assists in maintaining lip closure. The glossopharyngeal cranial nerve I and vagus cranial nerve X nerves manage many of the pharyngeal and laryngeal movements, and the hypoglossal nerve, cranial nerve II, guides the tongue's positioning. The transition from voluntary to involuntary control in swallowing is seamless. You begin the process consciously, deciding to swallow, and then the reflex pathways take over, freeing your attention for other tasks. This efficiency is particularly valuable when you are not consciously focused on eating, such as during conversation, or when taking in small sips of water without pausing to think about the mechanics. The epiglottis deserves special attention here. It is not a muscle but rather a cartilage structure that responds passively to the upward movement of the larynx during swallowing. Its leaf-like shape allows it to fold gently over the glottis, the opening to the airway ensuring that food and liquid are directed along the digestive route. Once swallowing is complete and the larynx lowers again, the epiglottis returns to its upright position, allowing air to flow freely into the lungs. In addition to mechanical protection, the nervous system is prepared to respond quickly if any material does enter the airway. The cough reflex is an immediate forceful expulsion of air designed to clear the trachea. While not part of the normal swallowing process, it serves as an important safeguard. In terms of function, the pharynx itself is not a site of digestion. No enzymes act here, and no absorption takes place. Its importance lies in its role as a safe, efficient passageway, guiding the bolus from the mouth into the esophagus without allowing it to stray into the respiratory system. For most people, swallowing is so automatic that its complexity is hidden. But when we slow down to observe it, we can appreciate the balance between speed and precision. The bolus is moved quickly enough to minimize the time the airway is closed, yet slowly enough for each protective measure to be in place. This quiet coordination is especially impressive when we consider that the pharynx must constantly alternate between two very different roles, carrying air for breathing and carrying food or liquid for digestion. Each time we swallow, the balance between these roles shifts for just a moment and then returns. Once the bolus has passed into the esophagus, the pharynx relaxes, the soft palate lowers, the larynx returns to its resting position, and breathing resumes. The airway is once again open, and the pharynx is ready to take in air or prepare for the next swallow. As we picture this process, it may be helpful to imagine the smooth passage of the bolus as it leaves the back of the tongue, moves through the oropharynx, passes the guarded entrance to the airway, and continues downward into the esophagus. There is no abrupt jolt, only the gentle pressure of muscles guiding it along the correct path. In summary, swallowing is a blend of conscious initiation and automatic reflexes, designed to move food efficiently while protecting the airway. The oropharynx and laryngopharynx serve as the transition zones, and the epiglottis provides a reliable safeguard. This combination of structures and reflexes ensures that, meal after meal, sip after sip, the journey from mouth to esophagus is safe and continuous. Now that the bolus has passed through the pharynx, we will follow it into the next part of its journey, the esophagus. Here, the role of movement continues, but with a different focus, the steady, wave-like motion of peristalsis carrying the bolus toward the stomach. Fechin's frave. From the pharynx, the bolus of food now passes into the esophagus, a long muscular tube that connects the throat to the stomach. It is a pathway of quiet efficiency, not a place of digestion, yet it plays a vital role in ensuring that each swallow reaches its destination. The esophagus is typically around 25 centimeters in length in adults, running behind the trachea and in front of the spine. It begins at the upper esophageal sphincter, located just below the pharynx, and ends at the lower esophageal sphincter, where it meets the stomach. For most of its journey, it lies within the chest cavity, before passing through a small opening in the diaphragm to enter the abdominal cavity. Although the esophagus may seem like a simple hollow tube, its wall is made up of 4 distinct layers, each contributing to its function. The mucosa is the innermost layer. Here, a lining of stratified squamous epithelium protects against mechanical wear as food passes through. Beneath it lies a thin layer called the lamina propria, and then a narrow band of smooth muscle known as the muscularis mucosae, which can create subtle movements in the lining. Surrounding the mucosa is the submucosa, a layer rich in connective tissue. It contains blood vessels, lymphatic vessels, and the submucosal plexus, part of the enteric nervous system which helps regulate secretions and blood flow. In the upper part of the esophagus, mucous glands within this layer release small amounts of lubrication, easing the passage of the bolus. Outside the submucosa is the muscularis externa, the true driving force behind esophageal movement. In the upper third of the esophagus, this muscle is mostly skeletal muscle, allowing voluntary control at the start of swallowing. In the middle third, the muscle is a mixture of skeletal and smooth muscle. In the lower third, it is entirely smooth muscle, controlled automatically by the autonomic nervous system. This gradual transition from voluntary to involuntary muscle reflects the shift from conscious initiation to reflex-driven transport. The outermost layer is the adventitia, a sheath of connective tissue that anchors the esophagus to surrounding structures in the neck and chest. In the short segment that lies within the abdominal cavity, this outer layer is instead a serosa, part of the peritoneal covering. The movement of food along the esophagus is not passive. It is guided by a series of peristaltic waves, rhythmic, coordinated contractions of the muscularis externa. Once the bolus has entered the upper esophagus, the muscles contract in a gentle sequence from top to bottom, creating a wave that pushes the bolus forward. This primary peristaltis usually moves the bolus into the stomach within a few seconds. If for any reason some material remains in the esophagus, a secondary peristaltic wave is triggered. This is not initiated by swallowing, but rather by the presence of the remaining food, ensuring that the esophagus is cleared completely. In both cases, the waves are unhurried but persistent, moving the bolus without relying on gravity. This is why it is possible to swallow even when lying down. At each end of the esophagus are sphincters that act as gates, opening when needed and closing to protect the pathway. The upper esophageal sphincter remains closed at rest, preventing air from entering the esophagus during breathing. It opens briefly during swallowing, then closes again to maintain separation between the respiratory and digestive tracts. The lower esophageal sphincter, also called the cardiac sphincter, is located where the esophagus meets the stomach. It is not a distinct anatomical ring, but rather a zone of specialized muscle that maintains constant tone. This sphincter opens to allow the bolus to enter the stomach, then closes to prevent gastric contents from flowing back into the esophagus. Its function is essential because the stomach contains acidic gastric juices that could irritate or damage the esophageal lining if reflux occurred regularly. The action of the esophagus is coordinated by both intrinsic and extrinsic nerve control. Within its walls, the myenteric plexus of the enteric nervous system manages much of the peristaltic rhythm. This is supported by input from the vagus nerve, which provides parasympathetic stimulation, and by sympathetic fibers that can modulate muscle tone. Although the esophagus does not chemically break down food or absorb nutrients, it is far from passive. It is an active transport system, ensuring that each swallow moves safely and efficiently from the mouth to the stomach. It works silently in the background, adapting its muscle type and coordination for the upper, middle, and lower sections, and maintaining constant readiness between swallows. When the lower esophageal sphincter opens, the bolus passes into the stomach. At that point, the esophagus relaxes and awaits the next swallow. Its role in the digestive sequence is brief but essential. Without its coordinated movement and protective gates, the rest of digestion could not proceed as smoothly. In summary, the esophagus, a muscular tube approximately 25 centimeters long, structured in 4 layers: mucosa, submucosa, muscularis externa, and adventitia, actively moving food via peristalsis, not relying on gravity. Guarded by upper and lower sphincters to maintain one-way flow, this quiet precision makes it an indispensable link between the mouth and the stomach. Now, with the bolus having passed through the lower esophageal sphincter, we arrive at the stomach, the next major site of digestion, where both mechanical churning and chemical breakdown begin. The bolus has now passed through the lower esophageal sphincter, leaving the esophagus behind. It enters the stomach, a hollow muscular organ that rests high in the abdominal cavity just beneath the diaphragm. Here the pace of digestion slows. The stomach serves as both a storage chamber and a mechanical blender, holding food until the small intestine is ready to receive it, while also beginning the process of breaking it down into a semi-liquid form. The stomach is shaped like a gentle curve. its broad surface pressed against the left side of the upper abdomen. It varies in size depending on its contents, expanding comfortably after a meal and relaxing back to a smaller shape when empty. Its wall is flexible, able to accommodate both small amounts of food and large meals without strain. Anatomically, the stomach is divided into 4 main regions, each with its own subtle role. Closest to the esophagus is the cardia, a narrow area that receives the swallowed bolus. It lies just below the lower esophageal sphincter, acting as the immediate entry point. The cardia produces mucus that helps protect the lining from the acidic gastric juices produced deeper in the stomach. Above the cardia and curving upward is the fundus. This rounded portion often contains a small pocket of swallowed air and plays a role in storing undigested food, allowing digestion to proceed gradually. The largest section is the body, or corpus. This is the central chamber where most mixing and secretion occur. The body of the stomach contains numerous glands that release acid, enzymes, and mucus, and it is here that much of the food begins to transform from solid particles into a softer mixture. At the far end of the stomach lies the pyloric region. This section narrows into the pyloric canal, which leads to the small intestine. The pyloric region is heavily involved in regulating how much of the stomach's contents pass forward, ensuring that the intestine receives manageable portions at a steady pace. The stomach wall is unique among digestive organs for its 3 layers of smooth muscle in the musculus externa. Most of the gastrointestinal tract has only 2, an inner circular layer and an outer longitudinal layer. The stomach, however, contains an additional inner oblique layer. This extra layer allows it to churn, twist, and knead its contents with remarkable efficiency. These layers contract in coordinated patterns, turning the stomach into a slow but powerful mixer. Food is pressed against the inner lining, broken apart mechanically, and combined with gastric secretions until it forms a thick, creamy substance known as chyme. The inner surface of the stomach is lined with rugae, folds of mucosa and submucosa that allow expansion after a meal. As the stomach fills, these folds flatten out, increasing the internal volume without significantly raising internal pressure. Beneath the surface, the stomach lining is equipped with numerous gastric glands. These are grouped into gastric pits that open onto the mucosal surface. Within these glands are different specialized cells, each releasing a component of gastric juice. Parietal cells secrete hydrochloric acid, or HCl. This acid lowers the pH of the stomach contents, creating an environment where certain enzymes can function optimally. The acidic conditions also help break down connective tissues in food and act as a barrier against many ingested microorganisms. Parietal cells also produce intrinsic factor, a glycoprotein essential for the absorption of vitamin B in the small intestine. Chief cells release pepsinogen, an inactive precursor of the enzyme pepsin. Once exposed to the acidic environment created by HCl, pepsinogen is converted into pepsin, which begins the chemical digestion of proteins, breaking them into smaller peptide chains. Mucus cells, also called foveolar cells, secrete a thick, alkaline mucus. This mucus coats the stomach's inner surface, forming a protective barrier that shields the epithelium from the corrosive effects of acid and digestive enzymes. Together, these secretions create gastric juice, a fluid that combines chemical digestion with protective functions. The process of secretion is regulated by both neural and hormonal signals. The sight, smell, and even thought of food can stimulate gastric glands through the cephalic phase of digestion, while the presence of food in the stomach triggers the gastric phase, intensifying secretion and muscular activity. Inside this environment, the food is gently mixed. Waves of muscular contraction move from the upper stomach toward the pylorus, grinding and blending the contents. The contractions are deliberate and rhythmic, creating a uniform texture and ensuring that every particle is coated with gastric juice. The pyloric sphincter stands at the junction between the stomach and the small intestine. This ring of smooth muscle remains closed most of the time, opening only briefly to allow small amounts of chyme to pass into the duodenum. By releasing chyme in controlled portions, the pyloric sphincter ensures that the small intestine is not overwhelmed and can efficiently continue digestion and absorption. The rate at which the stomach empties depends on the nature of its contents. Liquids pass through relatively quickly, while meals rich in fats or complex carbohydrates are released more slowly, allowing time for adequate mixing and breakdown. This careful pacing helps maintain a steady, balanced flow through the digestive tract. While the stomach is active in both mechanical and chemical digestion, it does not significantly absorb nutrients. A few substances such as water, alcohol, and certain medications can pass through the stomach lining, but most absorption occurs later in the small intestine. Throughout this process, the stomach maintains a balance between strong digestive activity and self-protection. The mucus barrier, rapid cell turnover, and tight regulation of acid secretion prevent damage to its own tissues. Disruption of these protective mechanisms can lead to conditions such as gastritis or peptic ulcers, but under normal circumstances, the stomach functions smoothly for many years without harm. To summarize, the stomach is divided into 4 regions: cardia, fundus, body, and pylorus. It has 3 layers of smooth muscle, allowing strong churning and mixing. Gastric glands secrete hydrochloric acid, pepsinogen, intrinsic factor, and mucus. Its primary functions are to store food, mix it into chyme, and release it gradually to the small intestine. From the calm storage of the fundus to the steady grinding of the pylorus, the stomach works patiently, preparing the meal for the next stage of digestion. Now, with the chyme ready and the pyloric sphincter opening in measured intervals, the journey continues into the small intestine, a place where digestion will reach its fullest complexity and absorption will begin. The chyme has now left the stomach, passing through the pyloric sphincter in gentle pulses. It enters the small intestine, the longest portion of the digestive tract, where the majority of nutrient absorption will take place. Though called small because of its narrow diameter, this section of the gut is far longer than the large intestine, stretching several meters in length. Here, digestion moves into its most intricate phase, one that combines the final stages of chemical breakdown with the careful uptake of nutrients into the body. The process is unhurried, designed to extract as much value as possible from the food we eat. The small intestine is divided into 3 regions: the duodenum, the jejunum, and the ileum. Each has its own structural features and primary functions, yet all share the same basic purpose: to process chyme thoroughly and pass nutrients into the circulation. The duodenum, a mixing chamber. The first portion is the duodenum, a short C-shaped segment measuring about 25 to 30 centimeters in length. It curves around the head of the pancreas and is firmly anchored in place, unlike the more mobile sections that follow. Here, chyme from the stomach is met by 2 important partners: bile from the liver and gallbladder, and digestive enzymes from the pancreas. These fluids enter the duodenum through a shared opening, the major duodenal papilla, regulated by a smooth muscle valve known as the sphincter of Oddi. Bile is a fluid containing bile salts, cholesterol, and waste pigments such as bilirubin. Its primary digestive role is to emulsify fats, breaking large fat droplets into much smaller ones, greatly increasing the surface area available for enzyme action. Pancreatic juice contains a range of enzymes: amylase to continue breaking down complex carbohydrates into smaller sugars, lipase to digest fats into fatty acids and glycerol, proteases such as trypsin and chymotrypsin to break proteins into peptides and amino acids. It also contains bicarbonate ions, which help neutralize the acidic chyme arriving from the stomach. This pH adjustment is essential, as most intestinal enzymes function best in a slightly alkaline environment. The duodenum is less about absorption and more about preparation. It is a true mixing chamber, ensuring that chyme, bile, and pancreatic secretions blend into a uniform mixture, ready for efficient absorption further along the tract. The jejunum, the absorptive heart. Beyond the duodenum lies the jejunum, which makes up roughly the next 2.5 meters of the small intestine. This section is suspended in the abdominal cavity by a thin, fan-shaped fold of tissue known as the mesentery, which also carries the blood vessels, lymphatics, and nerves that supply the intestine. The jejunum is the most active site of nutrient absorption. Its inner surface is highly specialized to increase surface area, giving more space for absorption to occur. This begins with circular folds, or plica circulares, large, permanent ridges of mucosa and submucosa that slow the passage of chyme and allow more time for digestion and absorption. Covering these folds are countless villi, small finger-like projections lined with epithelial cells. Each villus contains a capillary network and a central lymphatic vessel known as a lacteal. The capillaries absorb amino acids and simple sugars, carrying them directly into the bloodstream, while the lacteals absorb most dietary fats in the form of chylomicrons, which then travel through the lymphatic system before entering the circulation. On the surface of each epithelial cell, microvilli form what is known as the brush border. These microscopic extensions increase the surface area still further and also contain enzymes that complete the final steps of digestion. For example, disaccharidases break down double sugars into monosaccharides, and peptidases split small peptides into free amino acids. The combination of folds, villi, and microvilli increases the surface area of the small intestine by hundreds of times, ensuring maximum efficiency. Even though the chyme moves steadily forward, there is enough contact time for the majority of nutrients to be absorbed before it leaves the jejunum. The ileum, final absorption. The final and longest section of the small intestine is the ileum, measuring about 3.5 meters. Its structure is similar to the jejunum, but its villi are slightly shorter and more sparsely distributed. The ileum continues the work of absorption, focusing on nutrients that were not fully absorbed earlier, such as vitamin B12 and bile salts. Vitamin B12's absorption depends on intrinsic factor, the glycoprotein secreted by the stomach's parietal cells. In the ileum, intrinsic factor binds to receptors on the epithelial cells, allowing the vitamin to enter the bloodstream. The ileum also contains clusters of lymphoid tissue known as Peyer's patches. These form part of the gut-associated lymphoid tissue and play an important role in immune surveillance, helping protect the digestive tract from harmful bacteria. The transport of nutrients. Once absorbed, nutrients must be transported to the rest of the body. Water-soluble nutrients, including monosaccharides, amino acids, water-soluble vitamins and minerals, enter the capillaries within each villus. These drain into the hepatic portal vein, which carries the nutrient-rich blood directly to the liver. The liver acts as a processing center, storing some nutrients, modifying others, and releasing them into the circulation as needed. Fats follow a different route. Long-chain fatty acids and monoglycerides are packaged inside the epithelial cells into chylomicrons, tiny lipid-rich particles. These enter the lacteals and travel through the lymphatic system before eventually draining into the bloodstream via the thoracic duct. The pace of absorption. The movement of chyme through the small intestine is regulated by segmentation and peristalsis. Segmentation is a pattern of localized contractions that mix the contents and ensure they contact the absorptive surfaces. Peristalsis moves the chyme gradually toward the large intestine. The entire journey through the small intestine takes several hours depending on the type of meal consumed. This unhurried pace ensures that the body has ample time to extract nearly all available nutrients before the remaining material moves on. Maintaining balance. The small intestine is a delicate environment. Its epithelial cells are constantly renewed, with old cells shedding from the tips of villi and new cells arising from the crypts at their base. This turnover helps maintain a healthy absorptive surface and protects against injury. Digestive activity here is influenced by hormones such as secretin and cholecystokin, CCK. Secretin stimulates the pancreas to release bicarbonate-rich juice, while CCK triggers the gallbladder to contract and the pancreas to release enzyme-rich fluid. These signals help coordinate the activity of the stomach, pancreas, liver, and small intestine. Summary of the small intestine sept's role. To gently review, the duodenum mixes chyme with bile and pancreatic enzymes, neutralizing acidity and preparing for absorption. The jejunum is the main site of nutrient absorption, with extensive surface area created by folds, villi, and microvilli. The ileum completes absorption, especially of vitamin B12 and bile salts, and contributes to immune defense. Blood vessels and lymphatics transport absorbed nutrients to the liver or into the general circulation. Through patient-coordinated activity, the small intestine transforms the meal into energy and building blocks for the body. Its work is deliberate, methodical, and continuous, ensuring that nothing of value is wasted. From here, the remnants of digestion, mostly water, fiber, and indigestible substances, move onward to the large intestine, where the focus will shift from absorption of nutrients to absorption of water and to the preparation of waste for elimination. After the nutrients are drawn into the bloodstream from the small intestine, they take a very specific path, a path that leads directly to one of the most remarkable organs in the body, the liver. Positioned in the upper right quadrant of the abdominal cavity, just beneath the diaphragm, the liver is the largest internal organ. It rests quietly, filling its space with an unwavering presence. Every moment, without pause, it performs a vast range of chemical tasks that keep the body in balance. It does so without fanfare. No conscious sensation alerts us to its labor, and yet life depends on its continual work. Anatomy and structure. The liver is divided into two main lobes, the larger right lobe and the smaller left lobe, which are separated by a fold of connective tissue called the falciform ligament. Within these lobes, the liver's true functional units are much smaller, called lobules. Each lobule is a hexagonal arrangement of liver cells, or hepatocytes, organized around a central vein. At the corners of each lobule are structures known as portal triads, each containing a branch of the hepatic artery, a branch of the hepatic portal vein, and a bile duct. Blood from both the hepatic artery and the portal vein flows through a network of sinusoids, small channels lined with specialized cells, toward the central vein. Blood flow through the hepatic portal system. The liver is unique because it receives blood from two different sources. Oxygen-rich blood from the hepatic artery, which nourishes the liver's own tissues. Nutrient-rich blood from the hepatic portal vein, which carries materials absorbed from the small intestine. This portal blood is the first stop for most of the nutrients you absorb. It is here in the liver that these nutrients are processed, stored, altered, or released back into the circulation in carefully regulated amounts. The sinusoids, the liver's capillary-like spaces, allow close contact between blood and hepatocytes. Here, the cells can remove toxins, store glucose as glycogen, modify fats and proteins, and carry out countless other chemical adjustments. Bile production. Among its many functions, the liver produces bile, a greenish-yellow fluid essential for fat digestion. Bile contains bile salts, cholesterol, phospholipids, and waste products such as bilirubin. Its digestive role lies in emulsification, breaking large fat droplets into smaller ones. This increases the surface area for pancreatic lipase to act upon, making fat digestion much more efficient. The bile produced by hepatocytes drains into tiny channels called bile canaliculi, which merge into larger bile ducts. From here, bile flows either directly into the small intestine or, more often, is diverted to the gallbladder for storage until it is needed. Digestive and non-digestive roles. While its contribution to digestion through bile production is important, the liver's work extends far beyond this. It acts as a silent chemist, performing thousands of reactions that support life. Some of its roles include detoxification, removing harmful substances from the blood such as drugs, alcohol, and metabolic waste products. Nutrient storage, holding reserves of glycogen, fat-soluble vitamins A, D, E, K, and certain minerals. Synthesis of plasma proteins, such as albumin, which helps maintain fluid balance in the blood, and clotting factors, which are essential for wound healing. Metabolic regulation, converting excess carbohydrates and proteins into forms that can be stored or used for energy. Immune function, with Kupffer cells, specialized macrophages, removing bacteria and debris from the blood. Even though many of these tasks are not directly part of digestion, they are intimately connected to the way nutrients are used, stored, and kept safe. The liver as a regulator. The liver does not simply process nutrients, it regulates them. After a meal, when glucose levels rise in the blood, the liver takes in excess glucose and stores it as glycogen. Between meals, it breaks down glycogen to release glucose, keeping levels steady. When amino acids arrive from protein digestion, the liver can use them to build new proteins, or strip them of their nitrogen to create compounds the body can burn for energy. The nitrogen removed in this process is converted into urea, which is then excreted by the kidneys. Fats too are received, repackaged, and sent on to their destinations, some stored for later, some oxidized for immediate energy needs. This constant adjustment means that no matter when we eat or how much, the composition of our blood remains within the narrow ranges necessary for health. A quiet protector. One of the liver's most remarkable traits is its ability to regenerate. If part of the liver is damaged or removed, the remaining tissue can grow back to its original size, provided its structure is not too severely scarred. This regenerative power, combined with its detoxifying role, makes the liver a true guardian of the body's internal environment. Day and night, it stands between the outside world, with its fluctuations in diet, exposure, and toxins, and the sensitive tissues that depend on a stable internal chemistry. Integration with the digestive system. Though the liver is not physically part of the alimentary canal, its work is essential to digestion. Without bile, fats could not be absorbed efficiently. Without its metabolic regulation, the nutrients absorbed in the small intestine would overwhelm the body in sudden spikes and dangerous imbalances. The hepatic portal system ensures that everything absorbed in the intestines is first inspected and processed before entering the wider circulation. This arrangement is both protective and efficient, a central processing station through which all shipments must pass before being released. A steady presence. The liver works in silence, with no sensation to signal its constant activity. Its labor is rhythmic, steady, and dependable, a counterpoint to the more noticeable motions of chewing, swallowing, and peristalsis. If we could listen in, we would hear the soft rush of blood through sinusoids, the microscopic transfer of molecules between plasma and hepatocytes, and the slow trickle of bile into canaliculi. It is an organ of quiet chemistry, one that trusts the body to never notice, so it can continue its work undisturbed. Summary of the liverset's digestive role. Positioned beneath the diaphragm, the liver is the body's largest internal organ. It is composed of lobes and microscopic lobules, each with a central vein and surrounding portal triads. It receives both oxygenated blood from the hepatic artery and nutrient-rich blood from the hepatic portal vein. It produces bile, essential for fat emulsification, and performs numerous metabolic, storage, and detoxifying functions. It regulates nutrient levels in the blood, ensuring stability between meals. Through these roles, the liver acts as a constant overseer of internal chemistry, ensuring that what the digestive tract delivers is transformed, stored, or distributed with precision. From here, the bile it produces will meet the chyme in the small intestine, completing the digestive partnership between organ and tract. And the nutrients it processes will quietly fuel every cell of the body, long after the meal has ended. Tucked just beneath the right lobe of the liver lies a small pear-shaped organ, the gallbladder. It is easy to overlook in the vast arrangement of the abdominal cavity, yet its role is both precise and essential. While the liver is constantly producing bile, it is the gallbladder that holds this fluid in readiness, concentrating it until the moment it is needed. The gallbladder nestles into a shallow depression on the underside of the liver, connected to the biliary system by narrow ducts. Bile flows from the liver through these ducts in a slow trickle throughout the day. When digestion is quiet, most of it is diverted into the gallbladder. Here the fluid is stored in stillness, the walls of the organ gently drawing water out of it, concentrating its salts and pigments so that it will be more potent when the time comes to act. In this quiet reservoir, bile can become up to 10 times more concentrated than when it first left the hepatocytes. The release of bile from this storage space is not constant. It is carefully timed, in harmony with the arrival of food in the small intestine. When a meal containing fats enters the duodenum, a signal is sent in the form of a hormone called cholecystokinin. This signal reaches the gallbladder like a gentle tap on the shoulder, prompting its smooth muscle walls to contract slowly and steadily. The pressure inside the gallbladder rises, and bile begins to flow out through the cystic duct. The cystic duct merges with the common hepatic duct from the liver, forming the common bile duct, a shared channel that carries bile toward the duodenum. At the far end of this duct is the sphincter of Oddi, a small muscular valve that controls the entry of bile and pancreatic juice into the intestine. When the sphincter relaxes, bile slips through and meets the chyme waiting in the duodenum, spreading through it like dye through water. The emulsifying salts go to work, breaking fat droplets into smaller and smaller spheres, making them easier for lipase enzymes to reach. This interplay between gallbladder, ducts, and sphincter is an unhurried, measured process. It does not flood the intestine with bile all at once, but rather releases it in a slow, sustained manner, matching the pace of digestion. The bile moves and mingles with the chyme, transforming the texture of fats into a form that the intestinal lining can eventually absorb. When the gallbladder is not active, its walls rest in a soft folded state, the bile inside simply waiting. Its relationship with the liver is constant, the liver making, the gallbladder keeping. It is a partnership of supply and timing, ensuring that the small intestine receives what it needs, exactly when it is needed. Without this careful timing, fat digestion would be inefficient, and valuable calories and fat-soluble vitamins could pass through unclaimed. There is no sensation within the gallbladder that announces its work to us. No conscious awareness tells us when it contracts or relaxes. Yet every time a fatty meal is eaten, it quietly performs its part, its actions woven seamlessly into the choreography of digestion. Even when we sleep, if a late meal still lingers in the intestine, the gallbladder will respond, opening and releasing in the darkness. The bile it carries is the continuation of the liver's effort, a bridge between the chemical production deep in the hepatic lobules and the practical needs of the small intestine. Its store of concentrated bile is like a carefully measured reserve, never hurried, always deliberate. When the work is done and the gallbladder empties, it simply begins to fill again, awaiting the next call. In this way, the gallbladder's life is one of cycles: fill, hold, release, rest. A rhythm as dependable as the tides, set not by the clock but by the arrival of nourishment. And while its size is small, its role in the great sequence of digestion is as essential as any larger organ, for without it, the smooth digestion of fats would lose its balance and precision. From here, the story of digestion will turn to another organ that shares the bile duct's path to the small intestine, one whose secretions are equally vital: the pancreas, with its dual role as a digestive gland and metabolic regulator. Just beyond the curve of the duodenum rests the pancreas, a soft, elongated gland that blends almost invisibly into the surrounding tissues. Its position is quiet, but its influence is profound. Like a workshop that operates without noise, it produces substances that are essential for the breakdown and processing of the food we eat. The pancreas is a dual-function organ, Part of its work is endocrine, releasing hormones directly into the bloodstream to regulate blood sugar levels. This is the role of insulin and glucagon, hormones that maintain a steady balance between the storage and release of energy. Though this is vital to life, our focus here will rest more on the other half of its role, its exocrine function. This is the part of the pancreas dedicated to digestion, producing and delivering enzymes into the small intestine. The exocrine pancreas is composed of clusters of cells called acini. Each acinius contains specialized cells that secrete digestive enzymes into small ducts. These ducts merge into larger and larger channels, eventually joining into the main pancreatic duct. This duct runs the length of the pancreas, carrying its secretions toward the duodenum. In many people, the pancreatic duct meets the common bile duct just before it enters the duodenum, passing together through the sphincter of Oddi. In this way, the pancreas and the biliary system share not just an anatomical connection, but a moment of coordinated release. When partially digested food, now called chyme, moves from the stomach into the duodenum, it carries with it both nutrients and the strong acidity of gastric juice. The lining of the duodenum senses this arrival and responds by releasing hormones, primarily secretin and cholecystokinin. These signals reach the pancreas, prompting it to release its exocrine secretions. The pancreatic juice is a carefully balanced mixture. It contains enzymes for breaking down each of the major macronutrients. Lipase acts on fats, splitting triglycerides into glycerol and free fatty acids. Amylase targets carbohydrates, breaking complex starch molecules into smaller sugars. Proteases such as trypsin and chymotrypsin work on proteins, cutting the long chains of amino acids into shorter segments. These enzymes are produced in an inactive form, or zymogen, to protect the pancreas from digesting itself. Once they enter the small intestine, they are activated by specific chemical signals, beginning their work only where it is safe to do so. Alongside these enzymes, the pancreas secretes a significant amount of bicarbonate ions. Bicarbonate is alkaline, and its role is to neutralize the acidity of the chyme arriving from the stomach. This shift in pH is critical. It protects the delicate lining of the small intestine and creates an environment in which pancreatic enzymes can function optimally. Without this buffering action, the enzymes would not perform efficiently, and the intestinal tissues would be at risk of damage from prolonged exposure to acid. The flow of pancreatic juice into the duodenum is not abrupt. Like the release of bile from the gallbladder, it is gradual and sustained, matching the pace at which chyme moves through the intestine. This ensures that fats, carbohydrates, and proteins are steadily broken down into forms that the intestinal lining can absorb. The process is not hurried, but patient, allowing every molecule to be prepared for entry into the body's circulation. Even in its endocrine role, the pancreas works with the same quiet precision. Small clusters of cells known as the islets of Langerhans produce insulin and glucagon, releasing them into the bloodstream in exact amounts. These hormones help maintain stable blood glucose levels, ensuring that the energy extracted from food is stored or released according to the body's needs. Though this is a separate aspect of its work, it demonstrates the organ's dual nature, both a gland of digestion and a gland of metabolism. The pancreas does its work without sensation or signal to our conscious mind. We do not feel its enzymes being released, nor are we aware of its bicarbonate balancing the pH in our intestine. Yet its absence or failure would be immediately disruptive, halting the proper breakdown of food and depriving the body of the nutrients it needs. This quiet reliability is the mark of an organ that has evolved to serve its role with complete efficiency, without drawing attention to itself. As digestion moves forward, the pancreatic enzymes continue to interact with chyme along the length of the small intestine. Each passing minute brings further breakdown of fats into absorbable fatty acids, of proteins into amino acids, and of starches into simple sugars, together with bile from the gallbladder and the absorptive surface of the small intestine. The pancreas ensures that the transformation of food into usable components is both complete and orderly. By the time the chyme has passed far into the jejunum, much of the pancreas's role is complete. The enzymes have done their work, the bicarbonate has kept the environment stable, and the hormones have adjusted the body's internal chemistry to make use of the nutrients now entering the blood. The pancreas then returns to its resting state, awaiting the next signal, ready to begin again when another meal arrives. From here, our path through the digestive system will take us toward the large intestine, where the focus shifts from digestion to the careful recovery of water, electrolytes, and the management of the remnants that remain. From the narrow end of the small intestine, the journey of digestion enters its final stretch. Here, at the junction between the ileum and the large intestine, the body shifts its priorities. The rapid chemical breakdown of food slows, and the focus turns toward recovery, drawing water back into the body, reclaiming electrolytes, and preparing what remains for elimination. The large intestine is shorter than the small intestine, but broader in diameter. It begins with the cecum, a pouch-like chamber that receives material from the ileum through the ileocecal valve. This valve acts as a soft one-way gate, allowing the contents of the small intestine to enter, while preventing material from moving backward. Hanging from the cecum is the vermiform appendix, a narrow finger-like projection. Though its exact role is still studied, it is thought to participate in immune function and to serve as a reservoir for beneficial gut bacteria. From the cecum, the path ascends along the right side of the abdomen in the ascending colon, curves at the hepatic flexure, and travels across the body in the transverse colon. It then turns downward at the splenic flexure into the descending colon, continuing toward the sigmoid colon, a gently curved segment leading into the rectum. Each of these regions participates in the same essential tasks, but their arrangement allows for a slow, methodical progression of contents. The interior lining of the large intestine is specialized for absorption and for hosting vast communities of bacteria. By the time material enters from the small intestine, most nutrients have already been absorbed. What remains is a mixture of water, electrolytes, fiber, and undigested compounds. The colon reabsorbs most of the water and a portion of the electrolytes, such as sodium and chloride, returning them to the bloodstream. This process is steady and unhurried, ensuring that fluid balance in the body remains stable. The pace of movement in the large intestine reflects its purpose. Muscle contractions, called haustral churning, mix the contents gently, moving them from one pouch-like chamber to the next. Occasionally, stronger peristaltic waves, called mass movements, push material further along, often triggered by eating. Even so, the total journey from cecum to rectum may take many hours, or even a full day, allowing ample time for absorption and for the gradual transformation of the contents into feces. An essential feature of the large intestine is its microbiota, the immense population of bacteria that reside here in a balanced ecosystem. These microorganisms are not passive passengers. They actively ferment undigested carbohydrates, producing short-chain fatty acids that the body can absorb and use for energy. They also synthesize certain vitamins, most notably vitamin K, which is important for blood clotting, and some B vitamins. This quiet biochemical activity is continuous, shaping both digestion and overall health. The interaction between the microbiota and the host is one of mutual benefit. The bacteria receive a stable environment and a steady supply of nutrients from the fibers and other compounds that escape digestion in the small intestine. In return, they contribute to metabolic processes, support the immune system, and help prevent harmful microbes from taking hold. Though we cannot sense these activities directly, they form an integral part of the digestive system's daily work. The large intestine communicates closely with the nervous system to coordinate its movements. Stretch receptors in the walls detect the presence and volume of material, triggering reflexes that adjust muscle contractions. This communication ensures that the transit is neither too fast, which could cause excess fluid loss, nor too slow, which could result in excessive dryness of the feces. The balance is maintained quietly without conscious control unless we intentionally influence it through voluntary actions. As the colon absorbs water, the contents become progressively more solid. Feces take shape gradually, composed of undigested plant fibers, bacterial biomass, small amounts of mucus, and other waste products. The color and consistency are influenced by diet, hydration, and the state of the microbiota. By the time the material reaches the sigmoid colon, it is compact and ready for storage until elimination. The rectum serves as the temporary holding chamber. Its walls are sensitive to stretch, and when filled, they send signals to the brain that create the awareness of the need to defecate. The anal canal, the final segment, is guarded by two sphincters. The internal anal sphincter, under involuntary control, and the external anal sphincter, under voluntary control. Together, they ensure that elimination occurs at an appropriate time and place. When the body is ready, the process of defecation is coordinated between the rectum, anal canal, and surrounding muscles. This marks the conclusion of the digestive journey, a cycle that began with ingestion, moved through mechanical and chemical breakdown, absorbed essential nutrients, and ended with the removal of what could not be used. Even here, at the end of the process, the work of the large intestine reflects the same principles seen throughout the digestive tract: efficiency, balance, and quiet regulation. Its movements are steady, its absorption gradual, its interactions with bacteria constant but unseen. Nothing is rushed. Every step is given the time it needs. As we leave the large intestine, we can reflect on the continuity of the digestive system as a whole. From the mouth to this final passage, every region has played its part with precision. Each segment is specialized, yet all are connected by the shared purpose of sustaining the body. The large intestine closes the loop, ensuring that water is conserved, electrolytes are balanced, and the remnants of our meals are respectfully prepared for departure. From here, our journey will soon shift beyond the mechanical and chemical steps of digestion, moving toward the integration of this system with the rest of the body's physiology, the networks of circulation, metabolism, and control that make the digestive system more than a simple series of tubes and chambers. Beneath the surface of the digestive tract, hidden within the layers of its walls, lies a quiet network of intelligence. It does not think in words or images, yet it senses, decides, and acts with remarkable independence. This is the enteric nervous system, sometimes called the second brain. Unlike the brain in the skull, the enteric nervous system is spread out like a web, woven into the tissue of the esophagus, stomach, intestines, and beyond. It is composed of hundreds of millions of neurons, more than are found in the spinal cord, forming a system capable of regulating digestion without requiring constant instructions from the central nervous system. Its presence allows the gut to manage complex tasks, such as coordinating muscle contractions, adjusting enzyme secretion, and responding to local changes in pH or nutrient content, all on its own. The enteric nervous system resides primarily in two interconnected layers of nerve plexuses. The first is the myenteric plexus, or Auerbach's plexus, located between the circular and longitudinal layers of smooth muscle. Its primary role is to control motility, the timing, strength, and coordination of contractions that move material along the digestive tract. It senses when the gut wall is stretched, and in response, it can initiate waves of peristalsis, gently propelling food forward. The second is the submucosal plexus, or Meissner's plexus, lying just beneath the mucosa. This network focuses more on the inner environment of the gut. It regulates secretions, controls local blood flow, and monitors the chemical composition of the lumen. Through this layer, the gut can adjust its enzyme release, increase mucus production for protection, or alter absorption rates in response to the composition of what it contains. These two plexuses do not work in isolation. They are linked by countless connections, exchanging signals so that movement, secretion, and absorption are coordinated in a seamless flow. In this way, the enteric nervous system acts like a conductor of a silent orchestra, ensuring each section of the digestive tract plays its part at precisely the right moment. Though the enteric nervous system can function independently, it is also in constant conversation with the central nervous system. This connection occurs through the autonomic nervous system, the sympathetic and parasympathetic pathways. The vagus nerve, a major parasympathetic route, carries signals from the brain to the gut, often promoting digestion by enhancing motility and secretion. Sympathetic fibers, by contrast, tend to slow these activities, conserving energy for other demands during stress or exertion. Information flows in the opposite direction as well. Sensory neurons in the gut send messages back to the brain about its state, fullness, discomfort, or even subtle chemical changes. Many of these signals never rise to conscious awareness, but they influence mood, appetite, and overall well-being. This two-way communication is part of what researchers call the gut-brain axis, a partnership that extends beyond digestion into emotional and cognitive realms. Within the gut wall, the enteric nervous system relies on neurotransmitters much like those in the brain. It uses acetylcholine to stimulate contractions and secretions, nitric oxide to relax smooth muscle, and serotonin to coordinate peristalsis. In fact, the majority of the body's serotonin is found not in the brain but in the digestive tract, where it plays a vital role in regulating motility and sensation. The design of the enteric nervous system allows for highly localized control. For example, when a small segment of the intestines senses the presence of partially digested food, neurons in that region can trigger local contractions to mix the contents and stimulate glands to release enzymes, all without needing approval from higher centers. If another segment is empty, its neurons remain quiet, conserving energy. This autonomy ensures that digestion is tailored to the needs of each section, moment by moment. The sensory side of the system is equally remarkable. Neurons within the gut wall detect stretch, tension, temperature, and chemical signals from nutrients and metabolites. This information is integrated on the spot, producing immediate adjustments. It is a form of intelligence that does not rely on conscious thought, a continual, automatic adaptation to the ever-changing internal landscape. The enteric nervous system also plays a protective role. When harmful substances are detected, it can trigger strong muscular contractions to expel them rapidly, or increase fluid secretion to dilute and flush them out. These defense responses, though sometimes unpleasant, are crucial safeguards for the body. In the rhythm of everyday digestion, this second brain works quietly, rarely drawing attention to itself. It orchestrates the gentle waves that move food along, times the release of secretions with precision, and adjusts blood flow to match metabolic demands. It listens to the gut's internal world and to signals from the brain, blending them into coordinated action. Standing back, we can see that the enteric nervous system represents a deeper layer of digestive function, one that is neither purely mechanical nor purely chemical, but regulatory and integrative. It ensures that each step of the digestive process is aligned with the whole, and that the gut can continue its work even when the mind is elsewhere. To learn about it is to appreciate that digestion is not simply a sequence of passive reactions, but a living process guided by a complex and responsive network. The enteric nervous system is the unseen hand shaping the flow of digestion, moment by moment in harmony with the needs of the body. From here, our journey will shift again, outward, to see how digestion is supported by other systems of the body and how nutrients once absorbed are carried through the bloodstream to nourish every cell. As digestion in the small intestine quietly transforms food into absorbable molecules, another journey begins, one of transport. The particles that were once part of a meal now enter the body's internal waterways, carried along by currents of blood and lymph. It is a slow, deliberate voyage, as if following small rivers back toward a central reservoir before they are released to nourish the whole landscape of the body. For carbohydrates and proteins, the path begins in the tiny capillaries that weave through each villus of the small intestine. These capillaries are delicate, single-cell thick tubes, allowing sugars, amino acids, water-soluble vitamins, and minerals to pass easily from the absorptive cells of the intestinal lining into the bloodstream. From here, they do not scatter immediately to the rest of the body. Instead, they join a specialized route, the hepatic portal system. The hepatic portal vein is not a single solitary channel but a collection of merging streams. Blood from the intestines, pancreas, spleen, and parts of the stomach all converge into this large vessel, flowing steadily toward the liver. The journey is purposeful. Before nutrients enter the wider circulation, they first pass through the liver's gates, where they are processed, stored, or transformed according to the body's needs. In the liver's sinusoids, wide, porous capillaries, the incoming blood slows. Here hepatocytes, the liver's working cells, gently remove excess glucose and store it as glycogen, convert amino acids as needed, and filter out any harmful substances absorbed from the gut. Some nutrients are dispatched quickly into the systemic circulation, while others are kept in reserve to be released later when the body calls for them. This detour through the hepatic portal system is a safeguard and a point of regulation. It allows the liver to smooth out fluctuations in nutrient supply after a meal, ensuring that the body's internal environment remains steady. It is like a quiet customs station where every shipment is inspected before it moves on. Fats take a different route. Because lipids are not water-soluble, they cannot enter the capillaries directly. Instead, inside the intestinal villi, they are absorbed into specialized lymphatic vessels called lacteals. These lacteals form the beginning of the lymphatic pathway for dietary fats. Within the absorptive cells, triglycerides are reassembled and packaged into chylomicrons, tiny spheres of fat encased in protein. These chylomicrons slip into the lacteals, joining the clear, slow-moving fluid of lymph. The lymphatic system, unlike the circulatory system, has no central pump. Its flow is propelled by gentle contractions of surrounding muscles and by valves that prevent backflow. This means the journey of fats is unhurried, progressing steadily through larger lymphatic vessels toward the thoracic duct. At the base of the neck, the thoracic duct empties into the left subclavian vein, delivering the chylomins into the bloodstream. From here they travel to tissues such as adipose stores and muscle fibers, where they can be used for energy or reserved for future needs. In this way, digestion gives rise to two parallel transport networks. One, the blood route through the hepatic portal system, carrying water-soluble nutrients to the liver first. The other, the lymphatic path, carrying fats directly into the systemic circulation. Together they ensure that every molecule finds its correct destination. Even within the blood pathway, there is a quiet choreography. Capillaries in the intestinal villi merge into small veins, which in turn merge into larger veins, each carrying its share of nutrients toward the hepatic portal vein. Along the way, the smooth muscle walls of these vessels maintain a steady, gentle pressure, guiding the flow without haste. The lymphatic vessels, in parallel, weave alongside the blood vessels, collecting not only fats, but also excess fluid and waste from the tissues. Their contribution is subtle but essential, preventing fluid buildup and returning it to the bloodstream. When we think of digestion, we often picture the breakdown of food in the stomach and intestines. But this phase, the transport of nutrients, is equally vital. Without it, the chemical transformations of digestion would be like a harvest with no means of distribution. It is the movement through blood and lymph that allows the results of digestion to reach every cell in the body, from the tips of the toes to the neurons of the brain. And so, after the mechanical work of chewing, the chemical work of enzymatic breakdown, and the absorptive work of the intestinal lining, there comes this final act of delivery. It is quiet, continuous and far-reaching, connecting the inner surface of the gut to the outer reaches of the body. Seen from above, it resembles a watershed, countless small streams, the capillaries and lacteals, gathering into larger channels, then into main rivers, and finally joining the great circulation of the body's internal sea. This is the bloodstream and lymphatic system in digestion, the gentle couriers, the patient carriers, the silent link between the meal we have eaten and the energy we will soon feel. Beneath the surface of digestion, beyond the movements of muscles and the flows of enzymes, there is another layer of coordination, one carried not by nerves, but by chemical messengers drifting through the blood. These are the digestive hormones, each released in precise moments, each with a quiet influence that shapes the pace and rhythm of the entire tract. They work like unseen conductors, ensuring that the organs play their parts in harmony. One of the earliest to act is gastrin. This hormone is released by G cells in the lining of the stomach, especially in the antrum near the pylorus. Its signal is triggered when food enters the stomach, stretching its walls, and when proteins are sensed in the lumen. Gastrin's role is gentle but decisive. It encourages the parietal cells to release hydrochloric acid and stimulates the stomach's muscular layers to mix and churn. By increasing the stomach's secretions and activity, gastrin prepares the environment for protein breakdown, allowing pepsin to work more effectively. When the meal begins to pass onward into the small intestine, gastrin's influence recedes, like the first movement of a symphony yielding to the next. As the acidic chyme leaves the stomach and enters the duodenum, the balance of signals shifts. Here, secretin emerges, released by set cells in the duodenal lining. Secretin's message is one of moderation and protection. It senses the arrival of acidic contents and prompts the pancreas to release bicarbonate-rich fluid. This bicarbonate gently neutralizes the stomach acid, creating an environment in the small intestine where enzymes can work without harm. Secretin also encourages the liver to produce bile, ensuring that fat digestion will be well supported. It is a hormone of calm adjustment, smoothing the transition between the stomach's harsh chemistry and the small intestine's more delicate processes. Working alongside secretin is cholecystokinin, or CCK, released by eye cells in the duodenum and jejunum. Its presence is prompted by fats and partially digested proteins arriving from the stomach. CCK's influence spreads in several directions at once. It signals the gallbladder to contract, sending stored bile into the duodenum. It prompts the pancreas to secrete enzyme-rich fluid, and it slows gastric emptying, giving the small intestine time to process what has already arrived. CCK is like a careful timekeeper, ensuring that the pace of digestion matches the intestine's ability to absorb and assimilate nutrients. Other hormones join the orchestration in more subtle ways. Gastric inhibitory peptide, GIP for example, is released in the small intestine when fats and carbohydrates are present. It signals the stomach to slow its activity and at the same time prompts the pancreas to release insulin in anticipation of rising blood glucose. Motilin, secreted between meals, gently encourages the migrating motor complex, a wave of activity that sweeps through the stomach and small intestine, clearing away residual contents and preparing for the next meal. These hormones do not rush. Their effects unfold over minutes and hours, carried by the bloodstream from one organ to another. Unlike nerve impulses, which act instantly and fade quickly, hormonal signals linger just long enough to allow a complete, coordinated response. They are the digestive system's way of speaking in long sentences rather than short commands. What is striking is how these chemical messengers complement the enteric nervous system. The nerves within the gut wall handle rapid, localized adjustments, tightening a sphincter, increasing peristalsis, stimulating a nearby gland. The hormones, in contrast, coordinate activities across longer distances and over longer timescales. Together they form a dual system of regulation, one electrical, one chemical, ensuring that digestion is neither rushed nor delayed, but flows with steady precision. In this hormonal conversation there is constant feedback. When enough acid has been neutralized, secretin levels fall. When fats have been emulsified and absorbed, CCK release slows. When the stomach is empty, gastrin fades to silence. It is a system of balance, each hormone rising when needed, then withdrawing so that others can take their turn. The result is a digestion that feels seamless from the outside. We do not sense the release of gastrin when a meal begins, nor the quiet action of secretin as it buffers the duodenum. We do not notice CCK holding back the stomach while summoning bile and enzymes. And yet these hormones are always there, maintaining order in the background, letting us experience eating as something simple and satisfying, even though its inner workings are complex and finely timed. When viewed as a whole, hormonal regulation is like a slow-moving tide, subtle, pervasive, and essential. It ensures that every part of the digestive tract receives exactly the right message at exactly the right time, guiding the journey of food from the first bite to the last absorbed molecule. And as each hormone rises and falls, the body's inner rhythm continues unbroken, meal after meal, day after day. If the digestive tract is a river, then along its banks and within its depths lives a vast and bustling city, a community of life so numerous and diverse that its inhabitants outnumber our own human cells. This is the microbiome, a collection of trillions of microorganisms, bacteria, viruses, fungi, archaea, quietly sharing our space, and in many ways sharing our lives. Most of these companions live in the large intestine, where the slower pace and abundant nutrients create a perfect environment for settlement. But they are present throughout the tract, from the mouth's first welcoming surfaces to the folds of the colon. They form a shifting mosaic of species, each adapted to the chemical conditions, available nutrients, and physical landscape of its chosen niche. The relationship between host and microbiome is not one of simple coexistence. It is a partnership, a quiet exchange of benefits. We offer these microbes shelter and food. In return, they assist in processes that our own cells cannot perform. They help break down dietary fibers and resistant starches that escape digestion in the small intestine, fermenting them into short-chain fatty acids such as acetate, propionate, and butyrate. These compounds nourish the cells lining the colon, support a healthy intestinal environment, and even influence metabolism in distant organs. Some microbes produce vitamins, including vitamin K and certain B vitamins, adding their quiet contribution to our nutritional well-being. Others help regulate the balance of bile acids, influencing fat absorption and cholesterol metabolism. Still others participate in the transformation of plant compounds into bioactive molecules with subtle effects on health. All of this unfolds silently, without our awareness, as part of the daily rhythm of digestion. The microbiome is also an active partner in immune function. Its presence shapes the development of the gut's immune system from infancy, teaching it to distinguish friend from foe. A healthy microbial community discourages the growth of harmful species by occupying niches, competing for nutrients, and producing substances that limit pathogen survival. The gut lining and its immune cells are in constant conversation with these resident microbes, responding to their signals with adjustments that maintain a state of balanced tolerance. This balance is delicate. Changes in diet, illness, antibiotics, or stress can alter the composition of the microbiome, shifting the proportions of species and affecting its functions. When the community becomes disrupted, a state called dysbiosis, digestion may be impaired, immune regulation may falter, and the overall harmony of the system can be disturbed. But just as the microbiome can be disrupted, it can also recover, adapting and rebalancing with the return of favorable conditions. Diversity is its strength. A varied diet rich in plant fibers tends to foster a richer microbial ecosystem, with many species contributing different skills. In this way, the health of the microbiome and the health of the host are intertwined. What we choose to eat shapes not only our own nourishment, but also the well-being of the trillions of small lives within us. If we were to zoom in on this microscopic community, we would see a dynamic, shifting landscape. In one corner, bacteria are fermenting fibers into nourishing acids. In another, species are breaking down bile salts, recycling them for reuse. Elsewhere, microbes are processing polyphenols from plants, transforming them into compounds that can influence our own physiology. And in every part of this living city, there is constant communication, chemical signals drifting between microbes, and between microbes and the cells of the gut wall. This is not a static scene, but a slow, continuous dance of cooperation, competition, and adaptation. No single species dominates forever. Instead, populations rise and fall with the ebb and flow of available resources. Meals create pulses of nutrients that travel through the tract, and the microbiome shifts in response, adjusting its activity to match the rhythm of feeding and fasting. What is perhaps most humbling is that this vast community is not truly separate from us. Our cells, our immune system, our metabolism, all are influenced by the activities of the microbiome. In turn, the environment we create through diet, lifestyle, and health shapes the possibilities for the microbes that call us home. It is a partnership so deep that it blurs the boundaries of self. In quiet moments, it is worth pausing to imagine this hidden life within life, countless organisms, each with their own needs and cycles, yet all woven into the same larger system that sustains us. Every step of digestion, from the breakdown of complex carbohydrates to the absorption of nutrients, is subtly touched by their work. Their presence is constant, their activity unceasing, and their influence profound. When we think of digestion, we often picture organs, muscles, enzymes, and hormones, but the microbiome adds another layer, a living network of allies whose cooperation allows the digestive system to reach its full potential. It is a reminder that we are not solitary beings, but ecosystems, carrying within us a universe of life that thrives as we thrive. And so, as food moves through the tract and the body takes what it needs, the microbiome continues its work in the background, unseen but essential. This life within life is not hurried, not forceful but steady, a constant companion in the long journey from meal to nourishment. From the first scent of food to the final release of waste, the digestive system moves with a rhythm that is both ancient and constant. It is easy to think of each step in isolation, the mouth chewing, the stomach churning, the intestines absorbing, but in truth, every moment is part of a larger whole, a seamless flow that connects one organ to the next, and one process to another. Yet even this is only part of the story. The work of digestion does not end within its own walls. Its purpose is to sustain the body far beyond the digestive tract itself, to deliver the molecules that every cell depends on for life. Glucose from carbohydrates feeds our neurons, allowing thought and memory to take shape. Amino acids from proteins become the building blocks of muscle fibers, enzymes, and hormones. Fatty acids and glycerol from lipids provide both concentrated energy and the raw materials for cell membranes and signaling molecules. Vitamins and minerals, carefully absorbed, support hundreds of metabolic pathways. Iron carried away from the intestine becomes part of hemoglobin, transporting oxygen to every corner of the body. Calcium strengthens bones and also enables muscles, including the heart, to contract with precision. Water and electrolytes, reclaimed with care in the colon, help maintain blood volume, nerve conduction, and temperature balance. These resources travel through the bloodstream, and lymphatic system like provisions sent along winding roads to distant villages. Every tissue, skin, brain, heart, liver, bone receives what it needs when it needs it. The coordination of this distribution is quiet, without urgency, but it is constant. The digestive system's work is also deeply connected to the body's inner balance, or homeostasis. Through its interplay with the nervous system and hormones, it adjusts to our needs, slowing when the body rests, speeding when there is a demand for fuel, holding back when reserves are full. Signals from the stomach and intestines tell the brain when we have eaten enough, while hormonal messages influence how stored energy is used or saved. Even the microbiome, those trillions of unseen partners, plays a part in this integration, shaping metabolism, influencing the immune system, and sending chemical signals that may even affect mood and behavior. In this way, digestion is not simply a physical act, but a central thread in the body's interconnected web. The liver stands at a crossroads in this system, receiving nutrients from the small intestine through the hepatic portal vein. Here, molecules are processed, stored, modified, or released into circulation according to the body's needs. This processing is not hurried, it is deliberate, ensuring that the flow of nutrients matches the pace of the body's requirements. The pancreas, gallbladder, and stomach are not only servants of digestion but also guardians of energy supply. Their secretions, controlled by hormones and nerves, keep the transformation of food efficient and adaptable. If the body is an orchestra, the digestive system is both a section and a conductor, performing its part while guiding the tempo of energy availability. In times of health, this integration happens without our notice. We eat, the system responds, and our cells are nourished. In times of illness or imbalance, the connection becomes more visible. Poor digestion can ripple outward, affecting energy levels, immunity, and even mental clarity. This reminds us that while the digestive system asks little of our conscious attention, it benefits greatly from our care. That care can be simple. Foods rich in varied nutrients feed both our cells and our microbiome. Adequate hydration supports smooth movement through the tract. Periods of rest allow the system to reset, just as activity stimulates its flow. Gentle movement, like walking, can encourage digestion's rhythm, while mindful eating can give it the time it needs to work without strain. When we pause to consider the system as a whole, the image is not one of separate parts but of a living current, nutrients flowing in, being transformed, and moving outward to every cell. Each heartbeat sends digested molecules through the bloodstream. Each breath carries oxygen to meet them, and together they form the foundation of life's quiet continuity. It is humbling to think that while we move through our days, speaking, working, resting, this entire sequence unfolds without demand for our direction. The digestive system does not require us to manage its valves or measure its enzymes. It simply receives, processes, and delivers, sustaining life with a reliability that feels almost like devotion. In this way, digestion mirrors the natural rhythms around us, the tide moving in and out, the sun rising and setting, the seasons turning without haste. It is steady, persistent, and essential, yet rarely in the foreground of our awareness. Like these natural cycles, it works best when we align with it, respecting its pace and providing what it needs to flourish. And so, as we close this journey through the digestive tract, we see it not as a linear path from mouth to rectum, but as a circle of giving and receiving, food becoming energy, energy sustaining life, life enabling the search for food again. It is a rhythm we share with all living beings, a quiet reminder that nourishment is both a biological necessity and a deeply human experience. The body trusts the digestive system to work without pause. In turn, the digestive system trusts the body to provide the conditions it needs: balanced nourishment, moments of rest, and the gentle care that allows every cell to be fed. As the tide of digestion settles after each meal, the body enters a state of calm readiness, restored and prepared for whatever comes next. This is not an ending, but a pause in a cycle that will begin again with the next bite, the next sip, the next moment of nourishment.